Hello student, welcome to Anu's classes. Today in this video I am going to discuss some important question which is very much important for your upcoming SS first year final examination of Assam board. So the questions are: first question, mention the function of four holes of flower. So as you know that flower has four holes and these are sepals or calyx, petals or corolla, stamens or pistil. So function of calyx is to protect the inner part of flower. Function of corolla is to attract insect for pollination and protect the inner parts, while the stamens and pistils are the male and female reproductive organ. Next question, what is inflorescence and difference between racemose and cymose? The inflorescence is defined as the arrangement of cluster of flowers on a floral axis. Means how flowers are arranged on floral axis is known as inflorescence, and the difference are in racemose inflorescence it's so unlimited growth, whereas in cymose it's so limited growth. And in racemose, order of opening of flower is centripetal, whereas cymose the order of opening of flower is centrifugal. What is estivation? Estivation is the mode in which sepals or petals are arranged in floral bud with respect to other floral member. Means how the sepals and petals are arranged, that is called estivation. Estivation is of four type: valvate, twisted, imbricate and vexillary. So valvate means in this type of estivation the sepals and petals don't overlap but touch each other at their margin. This is the diagram. So in diagram, their margin only touch each other. Imbricate, in this type of estimation, the sepals and petals overlap each other in a non-specific direction. Here, the petals or sepals are overlap each other. Look here, one end is inside, other end is outside. Like that, every petals or sepals overlap each other. Twisted. Here, the sepals and petals overlap each other one specific direction. So one petals is completely overlapped and means it's come inside, and one is completely come outside. Vexillary, here one large petal overlap two lateral petals or wings and further overlap two smaller anterior petals or leaf. So this is the larger petal, it overlap lateral petal, then it overlap two smaller petal. So when you draw the diagram, no need to draw the diagram in below, just draw this diagram. I am just showing this diagram as you understand means clearly. Who proposed binomial nomenclature? Carolus Linnaeus, the famous scientist, proposed the binomial nomenclature in 1753. Binomial nomenclature, another name is binary nomenclature, and it is a system of naming species used for identifying species. Right, the importance of meiotic cell division. Meiotic is the important because it ensures that all living produced via sexual reproduction contain the correct number of chromosomes. Meiosis also produces genetic variation by way of the process of recombination. Next come for some important differentiation. First one is prokaryotic cell and eukaryotic cell. So this is the difference of prokaryotic cells and eukaryotic cell. So if difference come, you write like that. If short nose come, just you write one part as short nose. Next, Hartooth, Sapooth. So these are Hartooth and Sapooth. So these are the difference. So if 2 marks come, you just write 2 points. If 3 or 4 marks come, then you write 4 points. I am giving you 6 points. You can write any 4 points, whichever you feel comfortable. Active transport, passive transport. So the difference between active transport and passive transport. Next, xylem and phloem. This is the difference between xylem and phloem. You know the xylem and phloem are the main vascular bundles. C3 plant and C4 plant. Here 13 difference are given. So but no need to write 13 difference. You just remember 4 or 5, whichever you feel comfortable. Meristematic tissue and permanent tissue. So this is the difference. Meristematic tissue means cell divide, the tissue where cell divide repeatedly is called meristematic tissue. Permanent tissue means the cells are dead cell, means they do not divide. Write the structure and function of mitochondria. You know that mitochondria is known as powerhouse of cells. So this is the structure and function of mitochondria, which is one of the main important organ of our cell. Write the structure of chloroplast with diagram. So chloroplast, you know, chloroplast is found in all higher plants and it is oval and biconvex and it is double layer cell, or different parts of chloroplast are outer membrane, inner membrane, stroma, thylakoid and lamella. So outer membrane means it is the skin around the chloroplast which helps to protect it. The outer membrane is permeable. Inner membrane, it is added another layer of protection of the chloroplast. Stroma means fluid that is found within the chloroplast is called stroma. Thylakoid means these are the tiny disc shaped stack that contain the chlorophyll. It means within the chloroplast, these which contain the chlorophyll are known as thylakoid. Lamella, these are bridge-like structures of the chloroplast that keep the thylakoid spaced out, so that are called the lamella. So this is the diagram. This fluid is called lamella, and this is the outer membrane, this is the inner membrane. Then fluid sac means in between both the membrane there are some fluid are present, that is called fluid filled sac and chloroplast of the ribosome. And these like structure are called thylakoid, and these are called stroma. Liquid is called stroma, and these are called stroma lamella. The thylakoid are joined by some tube like structure which is known as stroma lamella, and these are called granum. Means single this is called thylakoid, but in group thylakoids are known as granum. So look here, the granum is giving here within a box, means single this is called thylakoid, and when it comes in group they are known as granum. Why mitochondria is called powerhouse of cell? Mitochondria are known as powerhouse of cell because it is responsible for extracting energy from the food through cellular respiration. Means it produces energy through cellular respiration. The energy is released in the form of ATP, and it is an energy currency of cell. Means it is the energy producing organ of cell. That's why it is called powerhouse of cell. What are the five kingdom classification? The living organisms are divided into five different kingdoms: Protista, fungi, plantae, animalia, and monera. What are the major characteristics of classification? So, on the basis of their characteristics such as cell structure, mode of nutrition, mode of reproduction, and body organization. These are the major characteristics. What are the different types of algae? Explain with example. There are mainly three different types of algae. Three different classes means chlorophyceae, rhodophyceae, pheophyceae. Chlorophyceae are known as green algae. Example is Spirogyra. Rhodophyceae is known as red algae. One best example is Porifera. Pheophyceae is brown algae. One best example is Laminaria. So you please remember the name. Chlorophyceae means green algae, and remember one example. Rhodophyceae means red algae. One example Porifera. Pheophyceae means brown algae. One example is Laminaria. And here I am giving what type of pigment is present. Also chlorophyceae, chlorophyll A, chlorophyll B is present. Rhodophyceae, pigment R, phycoerythrin is also present. And brown algae, here chlorophyll A, chlorophyll C, carotene, and xanthophyll are present. What is hydroponics? Explain advantage of hydroponics. Hydroponics means cultivation of plant without using soil. And advantage is conservation of water and nutrient, no more use of fertilizer, pesticide, and other chemicals. What is nucleopore? State their function. Nucleopores are tiny openings present in the nuclear membrane of nucleus. They are formed by fusion of two nuclear membrane. Function: this pore allow the movement of specific molecules between the nucleus of the cytoplasm. Explain different stages of Calvin cycle. Calvin cycle is divided into three phases. First phase is called carboxylation, where RUBP is converted to three carbon compound. The next phase is called reduction, where ATP and NADP is used for synthesis of sugar, and final stage is regeneration, where RUBP is generated for continuity of the cycle. So these questions are important for your upcoming final exam. In my next video, I am going to share some important questions which are very much important for your zoology section. Till then, thank you and goodbye.